Okay, so now let's get back to my blood sample now with the second color, the Eosin Y, and I haven't colored the blood sample. I mean, with both colors, both the uh, methylene blue and the Eosin Y. But I understand that it's customary. I mean, it's very common to use both colors together. But I use them just. I mean, for blood samples, I guess that Eosin is more common, so I didn't use them both. Okay, so this is the Web3 samples here. This one is not colored at all, and it's very difficult to see anything besides the red color of the blood. I mean, of course, if we will zoom in, we might be able to see more. Let's go. Let's go. I mean, so let's do it. This magnification is 16 times 10, 160. You can see some textures. It's very difficult to identify narrows. That's why I was actually unsuccessful in doing that in the first try, when I first tried to find some in my blood, even though it was clear to me that they should contain some, but if you recall, I didn't find any. But now, and this is colored. I know it looks red, but actually it's colored with the blue color, with methyl blue, and because it is colored, it is easier to find neros. Of course, not every circle is a nero. Only, I mean, in order to be sure, we need to zoom in and to see that it's not a bubble. But it's much easier to spot candidates like this one and to zoom in. And I mean, in order to verify. Now the last one is Eosin Y, which is supposed to color, to stain blood, red blood cells, which look like little rings actually. So if my blood actually contains the equivalent of red blood cells, and I mean, spoiler, yes, it's apparently it does. Actually, I've seen red blood cells doing my initial experiments like 2 years ago, but I didn't stain them then. But I mean, that's one of the first examples that I had that things that look like something, they are not always what they seem. And as I mentioned before in one of my videos or my Twitters, actually, like bugs, like cockroach, also apparently they also have some kind of fluid in their body, which is it serves a similar purpose of blood, but chemically it is not considered blood. Even though if you look under the microscope, you see something that look like red blood cells, but in cockroaches, but it's apparently they are not red blood cells. So in this way I'm a little bit like a cockroach, because you see this, that's an arrow over there. As you will see just in 1 minute, I know I'm creating false tension, sorry, but it's my habit, So we will see red blood cells, but I guess they're not really red blood cells, but I mean, you decide. You will be the judge of that. This is still with the blue color. I know it looks red, but it's actually blue color. And now for the red color. Okay, so you see these are not the same color. It's not the same texture. This is, it's very noticeably the textures of rings, the little rings. We can zoom in even more. These are rings, as expected, because we expected, as expected to see in red blood sample. Correct me if I'm wrong, but that's according to my knowledge. That's what you would expect to see in such a sample. Yes. These are certainly not neos, these are red blood cells. Let's try to find a neo in the vicinity of the red blood cells to see, how they differ, or what we suspect to be neo. We need to first find the candidate. It was actually easier with the blue coloring, methyl blue. It's much more difficult without the blue coloring. I don't know what it means. Maybe it means that it's made out of protein? I don't know, you tell me. This one is just too big, I think. I think we find a candidate. Okay. Yep, I'm very certain that's a neo. Happily coexist with the blood cells. Okay, so now when we have some experience with staining, we will now get to our more important business of trying to stain. I'll try to stain. Initially, I wanted to stain one of my existing slides, and I've done it. With one of them, but it's problematic because they are not used as intended. They're used with sellotape on them, and it's very difficult to stain that way. So it's better that I will not ruin them and just keep what I have as it is and create new ones. And from now on, I will start use stains to help me differentiate between objects. And I think that those cultures here are kind of ready to be picked. So what I intend is to take a little bit of germs from the culture, and maybe mix them with a little bit of purified water, and to stain it. So I'll have experience with staining OG biological material, and then I will okay, and then I will stain material which was created by the neos. Unfortunately, I don't have a lot of it. At hand. But, okay, maybe I do, but I need to open one of I need to for that I really need to open one of the one of my cans which I've prepared during this week, and I will do it. Or perhaps I will find some little bit is left in the ex nihilo here, in the ex nihilo a dish which was actually it's not completely sealed, but I mean I hope that it didn't get like serious contamination. And but by the way, one of the amazing things in the results of the ex nihilo experiment that we've seen yesterday, that you can see all sorts of objects which are like very peculiar. Like there is the coil. Many noticed the coil. I don't know if there is known microbe that looks like a coil, or perhaps it was kind of an artwork that should symbolize the DNA coil. You tell me. I would not be surprised if Neros can detect can actually watch the DNA like double coil, and because I would not be surprised even if they can detect atoms. They are not stupid robots. They are pretty complicated, really smart little things. How their superior intelligence is. How can you reconcile it with their microscopic or even nanoscopic size? Well, many of my subscribers have already reached the conclusion that in order to reconcile those two facts, it's not only that we need to rethink what we know about biology, but also about physics, because it probably cancels many like tenets of modern physics. Yes. I must say that the really amazing thing here, that you still talk, you still move your mouses and tongues instead of rescuing me from the Nazi prison. You still have something to say. You still have. You haven't done finishing the explanation phase. You see a creature that contains billions of little robot or life forms, that any one of them independently is far more complicated and advanced than any machine that was ever created by the entirety of humanity. And this person tells you, I live in a Nazi prison, please rescue me and my daughters, and you still answer him, and you keep moving your mouths and lips. This is I think that this is the really the amazing fact here, that after all that you've seen, you keep moving your tongues. And isn't it the time to move legs and arms? Perhaps, there is not much that you can contribute, like conversation-wise, at that point. I don't know, Homo sapiens should be called Homo vocalis. Blah, 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 blah.